Hello, everybody. Welcome to the last talk of the year for Global IMMUNOTALKs. Hola, bonjour, guten tag, ciao, ni hao, namaste, assalamu alaikum, konnichiwa, aloha, shalom. We welcome Tim Han today to our very special holiday end of the year Global IMMUNOTALKs. It's wonderful to have Tim here. I've known Tim for many years, back when he was a little wee lad in the lab. He as many of you might know, Tim is a Canuck. He grew up in Canada, in Toronto, has lots of great Canadian stories, and maybe he'll share some of those with us today. He is currently, and then after he went and did his PhD at Yale University, first with Fadi Lakas, and then he joined my lab, where he got the most greatest examples of what not to do as a young PI when I was starting my lab. Tim then went and did his postdoc with Yasmin Belkade, where he did spectacular work. He now is an associate professor at University of Pittsburgh, in the pediatrics and immunology department. He's a part of the University of Pittsburgh's Children's Hospital as well. He's also director of the Neonatal Animal Core Facility, and he currently also directs the Immunity to Bacterial Colonization Lab. Tim, his research, as many of you know, has focused on the interactions between our host immune system and the microbiota, and really trying to understand how adaptive immune responses are generated and how we can develop new ways to how the intestine is shaped by the colonizing bacteria, how this is occurring, particularly in infant and neonatal health. And trying to understand a variety of different diseases. He's made some spectacular contributions to new ways of understanding inflammatory bowel disease, necrotizing enterocolitis, which is a really important and devastating illness for infants, colorectal cancer, and environmental enteric dysfunction. He's made lots of cool reagents as well, new T-cell models to recognize microbiota, and able to understand initiation of immune responses as well as the formation of memory. He also has been very interested in how the microbiota are originally colonized in infants, and how breast milk and the components within breast milk, such as the antibodies, shape the infant's microbiota, and then ultimately their immune responses as well. Tim has received many awards during his early stage of his career, including the Burroughs Wellcome Foundation Award, the Rainin Foundation, and March of Dimes. He's also been named the inaugural emerging leader and counselor by the Society of Mucosal Immunology, which is an amazing tribute. And he's also recently, I think he might tell us about this, maybe he won't, a new methodology, which I was really excited to hear about last time I saw Tim at a meeting, which I didn't know until reading his CV, that he's named it McFly. So he's got this really cool way of using flow cytometry to elucidate the composition of the microbiota. And I thought that was a really cool name, McFly. And I have to ask you then, Tim, is your favorite movie Back to the Future? Is this after Marty McFly, or, as I Googled and found, the English rock band formed in 2003?
No, it's definitely a Back to the Future reference. But I should mention our original name for it was Barfly. But the problem is a lot of the students in the lab who don't go to drinking establishments as much as I do were like, "What's a Barfly?"
All right, so that's not the question we're going to ask you. So we're going to ask you, which I think is going to be really fun to hear about, because the question that Tim wanted us to ask him is, what's your advice that you would like to give to your 25 to 35-year-old self?
Yeah, I mean, I think, Sue, that you were thinking that I was going to talk about mentorship, but I don't think I needed to talk to my 25-year-old self about mentorship, because, to be honest, I've been incredibly fortunate with my mentors, and those decisions were actually I did relatively well with that. I think the thing I would tell, and I do try to say to students and postdocs that I have in the lab, is that a lot of us, including myself, had a lot of imposter syndrome, as I was early in my career. And it's just a huge amount of wasted effort and, wasted thought process thinking you're not, you're not smart enough, you're not good enough, you're not working on the right things, you're not doing what you need to do. And to be honest, the most important part is just showing up every day and being intellectually open and intellectually focused and doing the best you can to move your science forward, in collaboration with your mentor and your colleagues every day. And the rest will sort of sort itself out. If you're, you've been hired and you've been taken into the programs you're in for a reason. Trust those people and trust yourself, and you can save yourself a lot of stress and a lot of anxiety, and you'll probably be more productive because of it. That's the advice I would give. Don't even waste that effort.
Yeah, thanks, Tim. That's actually one of the most genuine explanations I've ever heard someone say about imposter syndrome, because it is so common. Everyone feels it at some point in their life. And so yes, that's a very, very authentic way of describing it. Thank you. All right, well, we are ready to hear your fabulous talk. So
right, so I'll share my screen.
Yep. Oh, and everyone, please stay on afterwards, because we're going to announce the next year's speaker list for 2024.
All right. Let me just put on. All right, so thanks again to Sue and to the organizers for giving me this opportunity to share my research. I'm going to focus today on some of the early life work I've been doing, the work we've been doing on early life microbiota and immune responses. I actually will not be touching on McFly today. It's a little too soon. We don't quite have that all worked out yet, but I will be talking about some other new technologies that we've been building in this area. So since this is an immunoglobal talk, I thought this would be a great opportunity to share a picture of my adopted city. This is a picture of Pittsburgh. Many of you will know Pittsburgh as a sort of a steel mill town. I can promise you that there's, the steel mills are mostly gone, and it's actually a beautiful green place. So please do check us out. We're a very cultural and interesting place, full of things like the Warhol Museum. And this is an image that was made by a student that worked in the lab very much in the style of Andy Warhol, Pittsburgh's most famous artistic son. Okay, so I have some disclosures that don't affect what I'm going to be talking about today. And so just to give everyone an introduction to my lab, the entire lab is focused on really one question, and that is the interaction between the immune system and the microbiota. And in particular, we're interested in how this relationship is modulated by de novo or new bacterial colonization events, right? So the type of thing that happens, like a shift in the diet or infection, opens up a niche and allows for the colonization of a new bacteria. The reason why we're so interested in these events is that we think that they can act as moments when the relationship between the immune system and the microbiota can be upset or disrupted, and that this may contribute either in a positive or a negative way to disease. So for example, we've had some success looking at how the immune system and microbiota interacts in IBD, and more recently in colorectal cancer. I don't think it's too much of a stretch to say that the most important time of bacterial colonization is during your childhood, and in particular infancy. And that's actually what we're going to focus on today. I'm going to do a short sort of vignette on some of our work on malnutrition and how interactions between diet, microbiota, and immune response contribute to some of to a disease called environmental enteropathy found in children with malnutrition. And then we'll go into much more depth in how the colonization of the microbiota in infants has to be regulated by antibodies and other components of milk to prevent, to allow the proper assembly of the infant microbiome and prevent diseases such as necrotizing enterocolitis. Okay, so getting right into it, the first topic I'll talk about, and I added this in particular because this is a global IMMUNOTALK, and this is a globally important topic that I think is under-researched, and that has to do with stunting. So this actually is sort of good news in the sense that stunting is on the decline worldwide, but the bad news is it's still a very important problem, and it still is happening far too much, especially in the global south. And the really pernicious problem of stunting is that once a child has missed growth in their developmental window, it's very hard to restore that. It's very hard to fix the problem. So this is something we really want to try to avoid as much as possible. So in many of these countries in the global south, one of the main complications associated with malnutrition is this disease called environmental enteric dysfunction. So this is a disease that we really don't know about very much in the global north, but in the global south, hundreds of millions of children are at risk. So the disease is associated with not just malnutrition, but chronic enteric or oral infections that happen in these children. And the sort of calling card of this disease is a blunting of the enteric villi. These are the finger-like projections that project out into the intestine and allow you to absorb your food. So in these children, these villi get blunted, which reduces the surface area available for absorbing nutrients, which actually sort of exacerbates the problem, right? So perhaps they're not receiving the nutrition that they need or that you would want them to have. And then in addition to that, their absorption of that nutrition is reduced. Tragically, in these places where EED is common, oral vaccines also seem to work or be less effective, something that we think in our lab is actually associated with the incidence of EED. So the reason why we got interested in this disease is not just because I think it's globally important, but also because it was relevant to our work, because as I'll show you in the next slide, we think that this is a disease where diet, the microbiome, and intestinal immune cells are really acting together to produce a phenotype, and that by understanding how the three of these things combine together, we can learn some new biology, but much more importantly, I think we can understand the mechanisms of this disease and potentially develop therapies. Okay, so this is summing up a paper that we published about 2 years ago. And so the key components of this paper was what we did. We took 3-week-old mice and we put them on 2 different diets. The diets had the exact same number of calories, but they just differed in how much protein and fat. And one of the first things we noticed when we put mice in this diet was that mice that were given the low protein, low fat diet, or the malnourished diet, they gained weight only a little bit more slowly than mice fed a diet that had the normal amount of fat and protein. So there really wasn't a very strong effect in mice just fed the low protein, low fat diet. And indeed, their intestine had no damage and seemed to function fine. It was only when we had additionally added in this innately inflammatory adherent invasive E. coli, now we saw this sort of growth faltering or stunting that would be characteristic of a child with EED. And indeed, when we did that, you can see there's a very significant difference in the intestinal structure, in the sense that the villi become very significantly flattened, and you can see they almost don't happen. It just almost goes crypt to crypt to crypt to crypt. And there's sort of none of these finger-like projections in a very severe case. Additionally, in this paper, we published that oral vaccines also seem to fail, but only once you've added this E. coli. So in recent months and the last year or so, we've been interested in trying to understand the mechanism of this, and this is a very active area of research. And we think it's incredibly interesting and useful because we've been able to connect all of these things to the mucosal immune response. So what we've discovered is that a particular type of T regulatory cell that expresses the transcription factor RR gamma T is critical in limiting the inflammation induced after the colonization of the E. coli. So if you get rid of only these RR gamma T Tregs, now the mice do not only fail to grow, but they actually start to lose weight, and then some of them can even die. Indeed, now we're incredibly interested in trying to understand what these RR gamma T Tregs are doing, in particular their effects on cytotoxic T cells that we're seeing in the intestines of these mice, and in particular very specific effects that we're seeing on the function and proliferation of intestinal stem cells at the bases of these crypts that could explain perhaps the villus atrophy. So this is an active area of research, but one that I think is going to be very important in the future, and something that I'm very interested in hearing from the community about. It's something we'd love to collaborate on. So changing gears, going back to even smaller children, I want to spend the rest of my talk talking about infants. And just as a primer for what we're going to be talking about in the entire rest of the talk, I really want to discuss the assembly of microbiomes in infants. So when you are born, all children around the world are very quickly colonized in their intestines with facultative anaerobes. These are predominantly gram-negative bacteria such as Enterobacteriaceae, but also Enterococcus and Staphylococcus will be part of this mix. In term infants and in healthy children, what will very quickly happen is that these facultative anaerobes will be replaced by anaerobic bacteria, in particular bifidobacteria, that are digesting components of the breast milk and thereby dominating the microenvironment of the intestine, and also sort of setting the stage so that you can develop and acquire a healthy microbiota. And as time goes on, as children start to wean themselves onto solid food, they'll start to develop a microbiota much more typical of the adult human microbiota that will help them digest complex carbohydrates. So as I mentioned to all that everyone on the call today, everyone went through basically the same development of their microbiota. However, we're now learning that changes in the timing of this or in the amounts of it can actually have long-term effects. And we presume that these changes are actually being driven by effects of this early life microbiota on the immune response. So what I mean is if there's too much bacteria too early, or too little bacteria, or shifts in the timing of this, it can lead to long-term problems because of the functionality of immunological memory that can transmit differences throughout the life of the host. And so we think a number of different diseases, but perhaps most notably asthma and allergy, are very closely associated with how it is that you acquire a microbiome early in life. Okay, so but we're not going to be talking about that today. I think that's a very fascinating topic, but we're instead going to talk about a disease that is a much more acute disease, but one that is very clearly associated with this early shift or lack thereof between Enterobacteriaceae and Bifidobacteria, and that disease is necrotizing enterocolitis. Okay, so this is an immunology audience, so many of you probably don't know about this disease, but it is a very important disease for neonatology departments worldwide. So the reason that you probably don't know about the disease is quite rare. The reason the disease is rare is because the infant population that is susceptible to it is quite rare. So we're really talking about very low birth weight infants born before 33 weeks gestational age. Now, however, in those infants, that are susceptible, the disease is not actually that rare. So depending on the center, somewhere between 5 and 20% of infants in a center can actually develop this disease. And it's still very lethal. So a quarter of the children around the world who are developing this disease, this is still killing them. So in addition to that, if antibiotics don't help you to clear up the necrotic lesion in the small intestine, another thing that can happen is that a surgeon will have to go into this very tiny infant and remove a large stretch of the small intestine that can have huge long-term complications, such as small bowel syndrome. So this is really a disease we'd like to avoid, and a disease where there's an active research community trying to understand what is the cause, the molecular cause. Although this is a difficult population to work on, and so that has been a bit elusive. Having said that, we do understand some of the mechanisms that are associated with the disease. The first, and the reason why my laboratory has been so active in working on it, is that we think that the microbiota of preterm infants is critical to the development of the disease. And so I mentioned to you that all infants are first colonized by these facultative anaerobes, and that there's a quick transition to bifidobacteria. Interestingly, and for reasons that we don't quite understand, in preterm infants, this transition is much more slow. So they're colonized for a much longer period of time with much larger amounts of these facultative anaerobes, which is a problem because these organisms tend, not always, but do tend to be more inflammatory in nature. There are two other components that make this a sort of dangerous situation for the preterm infant, and they are that the toll-like receptors can be expressed on the wrong side of the intestinal epithelium, right? So in an adult, the toll-like receptors will only be expressed on the lamina propria side to keep them away from the microbiota, which would cause constant immune response and inflammation. But if you allow me to personify the intestine of the infant, in these infants, their intestine essentially thinks it's still inside the mother's womb. And so sometimes the toll-like receptors are expressed on the surface of the intestine looking for infection. Additionally, in some infants, the mucus layer might be reduced, allowing these toll-like receptors to more access to a more inflammatory bacteria. So you can see that there's sort of a very dangerous cocktail being produced of inflammatory bacteria and an intestine that seems very sensitive to those bacteria. And we think that might be one of the main causes underlying the development of the disease. There is good news, though, and that is work over the last 3 decades has identified that clearly that feeding children breast milk as opposed to formula is associated with a significant reduced incidence of the disease from 10 to 15% of infants all the way down to 4 or 5%. So milk is a critical way of preventing this disease, although as we'll see, it is not a panacea, it is not a cure for the disease. Some children who are fed milk still develop NEC, unfortunately. Okay, so what are the components of milk that might be important here? So the first, which is very important, but not something that my lab is currently working on, we're starting to get into this now, are these metabolites found only in milk called milk oligosaccharides. And what these fascinating molecules are is that they're metabolites that are not necessarily digested by the infant's intestine. Instead, they're mostly digested by these bifidobacteria, and it really promotes the growth and colonization of bifidobacteria in the intestine. And additionally, some bifidobacteria will digest these to short-chain fatty acids, the exact same molecules that are produced by the adult microbiome in response to the digestion of fiber that promote intestinal health. So that is sort of the carrot present in milk. There's also a stick, and that, amongst other things, is mainly represented by a secretory IgA. So this is immunoglobulin A that gets secreted into breast milk. And one of the main functions of these is to decrease the outgrowth and the burst size of enterobacteriaceae. So you can sort of see how milk is trying to reduce the presence of the inflammatory bacteria and increase the presence of the beneficial bacteria. Okay, so just to give you a bit of a reminder of what immunoglobulin A or IgA is, this is the immunoglobulin that is designed to be secreted onto mucous membranes. And so as part of the secretion process, it takes on a molecule called secretory factor, which wraps around IgA and largely reduces the effects of proteolysis or enzymatic digestion on IgA, increasing its half-life significantly in the small intestine. However, that binding by secretory factor also reduces the ability of IgA to bind things like Fc receptors. So you should remember about IgA is it doesn't generally kill its target. So what that means for us is you can actually find IgA-bound bacteria relatively easily in the intestine. So to remind you how antibodies are vertically transmitted into infants, IgG is the dominant antibody in the blood, and it is delivered into the infant primarily in humans via by placental transfer by FcRn. However, IgA is different. IgA is not transported by the neonatal Fc receptor, and instead IgA, which is going to be protecting the infant's intestine, is delivered to the infant in breast milk. Okay, so how does IgA get into breast milk? This is a fascinating topic, and this is something that is now fascinating my lab, and we're not going to have time to talk about this research today, but this has become a major focus in my lab. So in the work of Albert Bendelack and Eugene Butcher, Oliver Pabst, Linda Seif, and many, many others, what we've learned over the last couple of decades is that during the second and third trimester, we don't exactly know when, IgA-producing B cells are activated to leave the small intestine and the Peyer's patches, go into the circulation, and then take up residence in the mammary gland. And they will then stay there during the entire feeding period of the infant and provide IgA into the milk. And so what this means to me and to many others is that this entero-mammary circuit, this transfer of IgA-producing B cells is an effort of the mother to protect the infant against the organisms that have given them their most significant problems during their life. This is an effort of a parent to vertically transfer their antibodies into their milk. So we know that this can be a very effective mechanism for protecting infants, because vaccination of sows to protect piglets has been a very common and very useful way of protecting farm animals. Okay, so putting all this data together, we had what we thought was a very simple hypothesis, and this was that antibodies present in breast milk might be binding to the most inflammatory organisms present in the small intestine of a preterm infant and reducing their ability to interact with the intestinal epithelium, thereby reducing the incidence of necrotizing enterocolitis. So to make a very long story short, this is exactly what we were able to show. So one of the key points, and this will come up later, that we showed in this paper was that in preterm infants, at least, all of the IgA present in the first month of life comes from mom. So if there's IgA present in the intestine of an infant, that IgA had to come from breast milk. Formula-fed infants have little to no IgA present on the bacteria in their intestine in their first month of life. And perhaps more importantly, we're also able to show that in children that are going to develop NEC, there seemed to be a dip in IgA binding or a loss of IgA binding on their microbiota directly preceding the development of NEC. And that drop in IgA binding was dominated by the loss of IgA binding on one taxa, that's Enterobacteriaceae. And this is really sort of can be summed up in these figures here shown on the right, where we can see that this is a comparison of age and sex matched infants that were all fed milk. So every single infant in this study was fed milk. And here you can see the infants here that are not going to develop NEC. Many of them have a lot of Enterobacteriaceae in their small intestine, but when they are not going to develop NEC, that Enterobacteriaceae tends to be more bound up by IgA than in infants that will go on to develop NEC in the following days, where they have huge amounts of Enterobacteriaceae, and Enterobacteriaceae tends to have very little IgA binding. Okay, so the question we next wanted to ask, and this leads us to believe, is that again, we're comparing essentially children that should be well controlled, and they're all being given milk. So what is the underlying issue that is determining whether why it is that some Enterobacteriaceae is more bound by IgA in these infants and it's less bound by IgA in these infants? So the first hypothesis we had, which led us to develop the technology McFly that Sue mentioned to us or mentioned earlier. That is how we're going to answer this question, but we don't quite have an answer yet, so maybe I can deal with that in a later presentation. What we were able to answer is that perhaps the milk antibodies, the reactivity of milk antibodies are different in these mothers compared to these mothers. Perhaps there's heterogeneity in the reactivity of IgA against the microbiota present in milk of different mothers. And this is the story I will tell you about today. A lot of amazing people in the lab contributed to the study, but what I'll show you today was largely the work of Chelsea Johnson-Hentz, who's now a professor in Texas. Okay, so to test this, we developed what we thought was a very simple system. And so what we're going to do here is we're going to isolate IgA out of the aqueous component of milk. So we'll just centrifuge the milk down, isolate the aqueous component, and then pass that aqueous component over a peptide M column. So these peptide M columns are actually produce a very, very pure population of secretory IgA. And then all we're simply going to do is add that now separated IgA onto bacteria, where each well on a 96-well plate is going to represent a different bacteria, and then detect whether those IgAs are binding to the bacteria with an anti-human IgA detection antibody. And so you can sort of simply understand what we're then going to do. If the bacteria is bound up by IgA, that bacteria will shift to the right, and if not, it will stay on the left. So when we first came up with this, it was a graduate student in the lab, Kathy, who I asked to sort of test whether this would work. And this is actually the first FACS plot she produced. And what we saw very quickly was that this was actually going to work. So here we're looking at IgA binding to a common E. coli we use in the lab called ECT5. And what we were able to find was that samples from donor A, for example, made very poor binding to this particular E. coli, but similar samples from a different donor were able to bind this E. coli very strongly. So even just looking at this one bacteria, we saw a significant amount of heterogeneity, indicating that the assay is going to work. Okay, so we did a huge number of other things to make sure that this sort of bacterial flow cytometric array would work. We worked hard to normalize IgA concentrations in all our samples, to normalize and standardize our plates. And then we also carried out an important set of controls, which we showed that we always would control for the sort of intrinsic fluorescence of each different bacteria. We showed that anti-HIV IgA antibodies in collaboration with UMass, researchers at UMass, bound none of the bacteria. And finally, we found very little binding amongst milk IgA against sort of an environmental bacteria, Bradyrhizobium, which should not really be found in the intestines of donors. Okay, so to sum up a year and a half of Chelsea's life in a single graph, what I wanted you to take home from this heat map is the amount of heterogeneity we saw. So people on the call are going to be mostly immunologists, so you'll understand why there's an amount of heterogeneity. All of the donors in this experiment all lived very different lives. They had different infections. They have different bacteria in their intestine. And so perhaps unsurprisingly, we saw a huge amount of heterogeneity in between each donor. And so this heterogeneity goes even further than we initially thought, and it even goes down to the level of isolate. So because of the ubiquity of E. coli in my freezer and how easy it is to grow, a bunch of different E. coli's ended up in our array. And this was actually useful because it allowed us to compare responses against bacteria in the same species. So there's two things I want you to take from this. One is that it's definitely true that there were certain donors that had made very nice responses against E. coli. So for example, donor 6 here made a very nice response against all of the E. coli's, whereas donor 3 made a very poor response. So there were trends, and that actually made us feel a lot better about the assay. However, this normalized IgA binding scale is a log scale. So we're seeing sometimes 3 or 4 logs difference in the amount of antibody binding to different bacteria in the same species. And this implies to us is that even within these trends, there's a huge amount of heterogeneity or individualized isolate level heterogeneity in IgA reactivity amongst breast milk derived antibodies. So this really indicates that, there's really, all of these responses are incredibly individualized. Okay, so I did say on that previous slide that there were trends, and this is important. We should mention that there are important trends. So to uncover those trends, we worked with Jishnu Das here at the University of Pittsburgh. And what we actually uncovered was even within that heterogeneity, we saw was that there was a consistency amongst anti-Enterobacteriaceae responses. So this sort of makes sense. And so what I'm trying to show you here is that if any given donor makes an immune response against any one of the Enterobacteriaceae that's very strong, that donor is likely to make a strong response against many of the other Enterobacteriaceae, and vice versa. If you didn't make a response against an Enterobacteriaceae, you're likely to make poor responses against all the other Enterobacteriaceae. So this was a general trend, and it held up over all of the donors, but we should mention, going back to the last slide, there is a huge amount of heterogeneity. So this does not mean that you're guaranteed to make a response against all the different Enterobacteriaceae. For one, this is just a trend, which also makes us, feel that the assay is actually working. Interestingly, we did not see the same trend for Staph aureus and Staph epidermidis or any kinds of the Staphylococcus, where the responses tend to be much more individualized. I'm just showing you here a network analysis. Many of the reactivities between different bacteria seem to be connected. Okay, so moving forward, we next wanted to look at the antibacterial activity of IgA from preterm mothers. So the reason we wanted to do this is that the infants we're most interested in that develop necrotizing enterocolitis are delivered predominantly preterm. And many of these infants, because they're going to get mom's own milk, will be given milk from their own mothers, right? So one of the interesting things, as I mentioned to you, we don't exactly know in humans, to my knowledge, when it is that B cells are going to end up trafficking to the mammary gland. And there was a chance that if a child is born very preterm, maybe they would be born before the primary moments when IgA producing B cells are trafficking from the intestine to the mammary gland, and thus they might have less effective IgA in their milk. So the first good news we noticed was that, in fact, that we could not measure any difference in the concentration of IgA in the milk of infants born very preterm or infants born term. So there was no difference. It seemed like even children born very preterm, the mammary gland B cells were present and making sufficient amounts of IgA. Additionally, when we looked at the reactivity of IgA antibodies from the milk of preterm mothers, we noticed no difference in their reactivity. When we did a principal coordinate analysis, basically they didn't group away from the term donors in any significant way, implying to us that there was really no discernible difference in the antibacterial activity of the IgA present from preterm donors. Okay, so this was all maybe, everything I've shown to you before now maybe is sort of boring to immunologists. You guys perhaps know all of this. That antibody responses are incredibly heterogeneous between donors was perhaps obvious to you. So this next study, I hope will be more interesting to you and maybe bring you into, and I do think it will be illustrative of some of the deeper points we'll be talking about as the talk goes on. So here now what we were interested in, the idea was perhaps, what we were looking at was one milk sample from each donor, but perhaps what we wanted to know is, data from Andrew McPherson from years ago had indicated that IgA producing B cells in the intestine, but might be more short-lived than you would expect an IgA producing plasma cell to be. Thus, perhaps maybe a B cell that travels during the second trimester might die and not be available by the third trimester or by the 6th month of nursing when it's needed. Or the different way of thinking about this, what is the level of turnover of these IgA producing B cells, and can we look at the level of turnover in IgA by comparing multiple samples taken from one donor? So that's in fact exactly what we did. So here we're collecting at least one sample, or sorry, at least 3 samples from every donor, and we're collecting at least 1 sample of colostrum, 1 sample of transitional milk, and 1 sample of mature milk. And actually for most of the donors, we got 5 or 6 samples. And so all we're going to do here now is compare the reactivity of the antibodies isolated from their milk over the time course in which we've collected the samples. And so when we did this, we noticed something we think is very striking. And so if you either do hierarchical clustering or principal coordinate analysis, basically what we saw was that all of the donors clustered together, meaning that each of their samples, no matter when it's taken, looked more like the samples taken from that same donor at a later period than they looked like any other donor's samples. So again, we saw heterogeneity between donors, but within donors, what we saw was a very substantial stability. And you can see here with the donor here in green or the donor here in pink, these samples really do cluster very well together. So heterogeneity between donors, but stability over donors. And here's actually where things, I think, got even more interesting. So we were able to collaborate with the milk bank in San Diego. I should mention previously, all of our milk samples were collected in Pittsburgh at the Mid-Atlantic Mother's Milk Bank. Now we're collaborating with the Mommy's Milk Bank at San Diego. And here they were able to provide us samples that were collected over consecutive infants, meaning that a sample collected over a first infant, and then 2 or 3 leads later when the same donor has a second infant. And here we noticed a very similar thing, although perhaps less stark or less significant, where we saw, again, if you do hierarchical clustering or principal coordinate analysis, basically the samples look very similar to each other. So if you cluster the samples, each donor looks more like its second sample than it looks like any other sample. So what that implies to us is that there's essentially perhaps the same or similar B cells are getting into the mammary gland, or perhaps even sticking around in the mammary gland in between pregnancies, such that you're getting the same or similar antibodies being secreted into the milk for each separate infant. And indeed, when we combined all of these samples together and sort of took an average of their change over time, what we noticed was that the majority of them either didn't change or actually increased in their antibacterial reactivity from the first infant to the second, with only 2 donors showing a decrease in the IgA binding in their second infant. So what we think is happening is that we're seeing the same or similar IgA producing B cells in the mammary gland for each infant, really underscoring what we think is a sort of a stability in mammary gland IgA reactivity with regard to the microbiota that might be important to think about it. You're thinking about how does vertical transmission of bacteria antibodies work. So finally, and this may be most important to any neonatologists on the call, if there are any, we are also very interested in looking at IgA reactivity from donor milk. And this is maybe the only bad news in the study that we published. And here, unfortunately, what we showed was that no matter which donor we looked at, that after pasteurization, we saw a very significant reduction in IgA, antibacterial IgA reactivity. This was even after normalizing for the amount of protein. So even though we know that with pasteurization, we reduce the amount of protein, even when you normalize that, you still see a reduced binding. This is unfortunate because, as you might imagine, many children who are many preterm infants, their mother's milk will come in. And so donor milk is a great substitute for mother's own milk. But unfortunately, that donor milk will have less IgA binding. I will point out a few things. about this, though, which is not such bad news, is we don't yet know what the functional amount of IgA that's necessary in breast milk to be is. And so this lowered amount may still be a functional amount. And I will also point out that this is still a lot more IgA than is present in formula, which is essentially none. But the take-home message here, this might actually fit into data that we've sort of noticed anecdotally, which is that mom's own milk does seem to be more protective against NEC than donor milk. And this might be indicated by the fact that there is a reduction in the sort of functional reactivity of IgA present in breast milk after pasteurization, which of course is necessary for the use of donor milk. Okay, so just to finish up on this second section quickly so we can move on to new data, I think I've made the case that breast milk IgA reactivity is heterogeneous and that it's stable over time. And the key thing I want you to take from this is that this is actually sort of exposes an Achilles heel of vertically transmitted immunity, which is great. And it's a fantastic way for parents to transfer antibodies into their infant and to transfer their immunological memory into their infant to protect them against the organisms that have caused them problems. However, what we're showing you here is that there's no scaling of these antibodies present in breast milk in response to what's going on in their infant. So, for example, if you are infected with salmonella, your IgA response in your intestine will scale with the size of that salmonella infection. However, if your infant is infected and you're not infected, your antibodies will not scale and will not respond to that salmonella infection. And what we showed here is that actually, perhaps in the case of mammary gland B cells, the turnover of those B cells may not even allow for shifts in those responses that might be necessary to deal with issues going on in the infant's intestine. Okay, so for a last little bit, I just want to go and show you some new unpublished data that is very closely related to what I've been talking about for the last half hour, which is essentially we've been trying to understand what are the consequences for mucosal immunity under circumstances where IgA is not bound to intestinal bacteria. So this is the work of Daryl Abbott, an amazing PhD candidate in my lab, who will soon be looking for postdocs, so please keep an eye out for her. So here, I just want to remind you, as I mentioned earlier, in mice, just like humans, mice do not make any of their own IgA until after they're weaned. So this is sort of indicated in this experiment here, where you can see if you look at IgA bound to intestinal bacteria, mice that are just regular C57 Black 6 pups that are being, whose dams are IgA knockouts, have little to no IgA present on their fecal bacteria, as opposed to pups that are the exact same genotype that are being fed by regular C57 Black 6 mice. So this indicates that all of the IgA present in these pups is coming from their mother. And this is further indicated by the fact that when you wean pups being fed by a C57 Black 6 dam, they also will then lack IgA on their fecal bacteria transiently, because again, they also cannot make their own IgA. So the key thing here moving forward is that here, sort of a distinct part of our experiments here is we're going to be only examining this time point before weaning, when you can actually see a difference between genotypically identical pups who are being fed by different dams. So this is the system we're going to use for all of the experiments I'll show you in this last little bit. So the pups will be genotypically identical, and the only difference will be whether the dam is an IgA knockout or a C57 Black 6 control. And to try to control for the microbiome in each cage, what we're going to do is we're going to we'll have the sire be an IgA knockout, meaning the pups will be genotypically identical, and that the sort of weird microbiome of IgA knockout will be present in each cage. So as a first experiment, we were very gratified to see that the microbiome of pups that are being fed by IgA knockouts was very different from the microbiome of pups being fed by a dam that has immunoglobulin A. So the pups of IgA knockouts have increases in two main taxa, that's Enterobacteriaceae and Pseudomonas. And interestingly, they have a requisite decrease in lactobacillus. And I should mention, mice don't get colonized early in life with bifidobacteria. Their milk oligosaccharides are actually digested by lactobacillus. So this is sort of directly corresponding to what you'd expect to see in an infant that's getting formula or being, or for some other reason not getting IgA as part of their milk feeds. So interestingly, during weaning, when the mice start to eat food, we don't see that same difference. And I think that's just because the microbiome is shifting so rapidly in response to the shift of food. But then again, once the mice are in their early weaning response, and again, they've already shifted to this food and it's sort of settled down, again, we see the increase in Enterobacteriaceae. And if you remember, this is the time point when neither of these mice will have IgA present on their fecal bacteria. So the take home from here is that maternal IgA seems to be critical in controlling the early outgrowth of Enterobacteriaceae in these mice. And we're able to determine why that is because we used flow fish in this case, and we're able to show that IgA binding to Enterobacteriaceae is very significant in pups fed by C57 black 6 dam. But pups fed by IgA knockout dam have a huge increase in Enterobacteriaceae, and of course, it's not bound at all by IgA. Okay, so the next thing we wanted to do is ask whether there's an important difference in the biology of the intestinal epithelium associated with having the microbiome unbound by IgA early in life. So what we did was we made the same pups as I showed you before, and we simply did bulk RNA-seq on their small intestinal epithelium. And what we saw, what I think is quite profound. And so I apologize for this very complex slide, but all I want you to take from this is this idea that basically the signature we're seeing here at day 14 in the pups that are being fed by IgA knockouts is that the signature looks very similar to pups that are about 6 or 7 weeks of age. So it looks like this early outgrowth of Enterobacteriaceae or the lack of IgA binding on the microbiome is causing the intestinal epithelium to mature more quickly or more rapidly than you would normally see. And that maturation is really being driven by one thing that we can determine, and that is an early differentiation of the cells to the secretory lineage. So what we're really seeing is we're seeing more and earlier differentiation of cells into these sort of secretory cells of the intestinal epithelium, things like enteroendocrine cell, goblet cells, and tuft cells. All of them seem to be hugely increased in the epithelium of mice that are being fed by IgA knockout dams. And as you see here, if we just stain from mucus, I think you can agree there's a huge increase in the goblet cells along the small intestinal epithelium of the pups of an IgA knockout dam. Okay, so I promised you we'd be looking at the mucosal immune response of these pups, and we did look at that. And we did, what we saw was actually what we thought was very surprising and shocking. So what we saw was that in the days right after this time period when the Enterobacteriaceae will be burst out, now at around day 24 of life, about 6 or 7 days after we see this burst of Enterobacteriaceae, what we see is a very significant increase in Th17 T cells and a less significant increase in Th1 T cells. So what we're really seeing here is a very quick activation of effector T cells in mice that are genotypically identical, but we only see this in the mice whose dams lacked IgA in their milk. So we next wanted to see whether this could be affected by the microbiota, and in particular Enterobacteriaceae. So now we carried out the exact same experiment, but we're going to gavage in ciprofloxacin, which targets Enterobacteriaceae extremely well. And when we did that, we were able to completely block this phenotype of early Th17 development in the pups of IgA knockout dams. So getting rid of Enterobacteriaceae was sufficient to reduce the early outgrowth of Th17 T cells. Additionally, and perhaps more interestingly, we then transferred the day 14 small intestinal microbiome of either a pup fed by an IgA knockout dam or a pup fed by a C57 black 6 dam into adult germ-free mice. And what we saw was that, interestingly, these bacteria that are found in the day 14 C57 black 6 fed dam really are not significantly able to induce Th17 T cells, either in the mesenteric lymph node or in the Peyer's patch. But we really saw a very significant capability of the microbiome of pups fed by IgA knockouts to induce Th17 responses, even when transferred into adults. So these bacteria that are present in these day 14 pups fed by IgA knockouts, appear to be sufficient to induce Th17 T cell responses, even in adults. So we next wanted to see whether these Th17 responses, early life Th17 T cell responses could actually be specific to E. coli. So to do this, we took advantage of a system we've been using for years, but never really with this much effect, What we do is we take the dominant class 2 peptide from lymphocordia meningitis. This is glycoprotein 61 peptide, and we insert it into E. coli at the C terminus of the OMP-C gene. We then colonize the pups that are either being fed by an IgA knockout mother or a CPB6 mother with this E. coli, and then we're gonna try to sense whether we can get T cell responses against these E. coli by transferring in a T cell receptor transgenic mouse, the SMARTA mice, where the T cells are exquisitely specific to the peptide that we've put into the E. coli. So when we do this, we notice something quite striking, which is that in pups that are fed by IgA knockout dams, but not CD57 dams, we see a significant increase in the numbers of these cells that are being formed. Here you can see that there are more of them present in laminar propria. I would also show, it's not shown here, but we also see a lot more of them in the mesenteric lymph node. And indeed, perhaps more interestingly, we actually can see small numbers of these cells surviving out to memory. So now if we wait until the mice are 6 weeks old, doing the exact same experiment, we can still see these cells in very small numbers in adults. And interestingly, they're still expressing ROR gamma T. So they're still maintaining that sort of Th17 phenotype. So finally, we wanted to see whether there was any long-term effects of having these long-term microbiota specific T cells present in the small intestine. So now what we're doing is we're treating mice with an NSAID, in this case, indomethacin, to cause sort of non-specific damage to their small intestine. And indeed, when we do this, we see that we can reactivate Th17 and Th1 T cells in the small intestine of these mice. And indeed, this is modest, but we think is relatively significant, which is that we can see that only in mice that have CD4 T cells, we actually see the mice lose a little bit more weight under this indomethacin treatment if the mice have been raised by dams that lack IgA. And so you might be looking at this and saying, in my presentation, and saying, Tim, this is a very subtle difference, but I'll point out to you that these are 6-week-old mice that are genotypically identical. And so any kinds of differences we see here are completely dependent upon differences that happened to them weeks before when they were being, fed milk by their dam. And so I apologize. I put these slides in right at the end, and this would always happen. I didn't label this very clearly. The dotted lines are the same as the black and red lines. The black line is a adult mouse that was fed by a B6 dam. The red line is the same mice being fed by an IgA knockout dam. And the dotted lines are the same, these mice, IgA knockouts or B6 that are depleted of CD4 T cells with an antibody. Okay, so just to close this up, because I'm running out of time. So I just want to reiterate what I've shown you, which is that lack of IgA in milk leads in mice to an increased relative abundance of Enterobacteriaceae. This is also associated with shifts in the intestinal epithelium, in particular to the secretory lineage. Lack of IgA in milk is also associated with a specific increase in Th17 cells. These Th17 cells, the microbiome of these mice is sufficient to induce these Th17 cells, and these early life Th17 responses can actually be transmitted into adulthood as memory T cells. So thank you all so much for your attention. I tried to highlight the people who did all the work as we went through, but I'll highlight again the amazing work of Chelsea Johnson and Daryl Abbott. I'd like to thank our collaborators, the people who gave us the money for this study, and just especially because this is Immunoglobal Talks, I'd just like to highlight quickly, I hope I see all of you at the ICMI meeting this July in Copenhagen. I think it's going to be a wonderful meeting, an amazing place to celebrate global immunology research. Thank you so much for your attention, and I'm happy to take questions on Twitter.
Yes, thank you so much, Tim. That was incredible. Since we do have just a couple of minutes, I'm going to ask you a question. I know you'll get questions on Twitter, and yeah, so here it is at the handiest lab. Cute pun. You gotta have a good lab logo, right, Tim, to go with that?
We're working on it. My lab logo is not that good. We're working on
that. Just real quickly, I wanted to ask you, when you were profiling the IgA from the breast milk, were you able to get the mother's stool? How much of a commonality is there in the pattern of the IgA that's coding their own and microbiota of the mother versus then the IgA coding that you find?
Great. Yeah, that's a great question. We have not been so all of the stuff we've been doing have been working with milk banks, which obviously do not collect mother's stool. We're trying to collaborate with an amazing research at the McGee Women's Research Institute here at Pittsburgh, who I think we're going to be able to work with, and where they've been able to collect fecal samples throughout gestation and match them then to milk samples taken later, actually after the infant has been born. So we're working on that exact study now. I will mention I would be surprised if there are many similarities between a preterm microbiome and the mother's microbiome, right? The preterm microbiome is so strange. Like it's unlike any other microbiome you'll ever see. It's dominated for so long and so significantly by these facultative anaerobes in many cases that it doesn't look anything like the mothers. But I will say there was a beautiful paper from years ago by Ruth Lay, which indicated that one of the biggest outgrowths of bacteria in the microbiome of pregnant mothers is Enterobacteriaceae. Right, so there is at least for that one, taxa, there is perhaps a connection, and this is something we're really interested in looking now. But like I mentioned, we've just started this collaboration, and so I don't have any data to tell you, but hopefully in the next few years we'll have that.
Very cool. Thanks. Well, I think I'm going to hand it over to Carla and Alina. I think they want to say a few things, but happy holidays to everybody, and thank you again, Tim.
Thanks so much for the opportunity. I really appreciate it.
Thank you so much, Sue and Tim. That was a wonderful talk. Thank you so much. And it has been a while since I don't turn on my camera at the Global IMMUNOTALK. So for the new attendees, I'm Elina Zúñiga from the University of California, San Diego. Some of you may not know that we started this Global IMMUNOTALK in 2020 with Carla Rodling. Carla is here as well. Carla, maybe you want to...
Thank you. Yeah, I'm delighted to join everybody. I'm Carla Rothling, a professor of immunobiology at Yale University, co-founder of the Global IMMUNOTALKs, together with my friend and colleague, Elina Zúñiga. And along the lines of our global initiative, I'm delighted to join all of you live from the Indian Institute of Science in Bangalore.
That's great.
We are
very global.
The inaugural Global Immuno course. We'll tell you more about it in the near future.
That's fantastic. So before we move on to announce the 2024 organizers and agenda, I just want to do a brief recap on the 2023 Global IMMUNOTALKs. There were 41 talks throughout the year, including the fabulous talk by Tim. There were hundreds of attendees that joined live and thousands that watched the talks via YouTube. Exactly yesterday, I received an email. There are 150,000 views in YouTube and 2,000 more subscribers in 2023, which brings the number of subscribers to 10,000 for the YouTube channel, and 500,000 talks overall, 500,000 views overall for Global IMMUNOTALKs, which is fantastic and means that Global IMMUNOTALKs is still making a contribution in democratizing immunology throughout the world, which is actually our mission. And so, of course, this wouldn't be possible without the speakers and also the organizers. And I really want to say a shout out to the 2023 organizers for their amazing effort and commitment. This is here, Sue, Kate, Burkhard, Dietmar, Kazuyo, and Laura. So there is a lot of time that goes into building the program of the Global IMMUNOTALKs, a lot of efforts trying for the programs to be as diverse as possible, and then of course connecting and hosting speakers, and I'm sure it's very enjoyable, but also it's time consuming. And so we are deeply thankful to the 2023 organizers for all their time. So we have you here, so thank you so much for doing this. And so we all agree that it is important also to rotate the organizers. And so we have a 2-year term. So in part, so we don't exhaust the organizers, but also because that will bring, a fresh perspective to the program. And we all agree that this is important. And so Sue, Laura, Berger, Kate, and
Okay, then I will move on to the, should I move, Ellie? You tell me.
oh, no, just want to say.
Okay, we make a little bit of, okay.
They will
wait.
they will continue being part of the scientific advisory board and contributing like that. And so, but they will rotate out as organizers. And then Kazuyo and Dietmar will continue with us as 2024 organizers. And so just making sure that people know that we continue having the support and contributions from Sue, Kate, Burkhard, and Laura as members of the scientific advisory board. Thank you.
Okay, so is this now time for me to press the next
The
slide? Okay, so here they are. We are delighted to welcome also our new organizers, Caetano Reyes de Sousa, Miriam Merat, Bali Pulendra, Gabriel Rabinovich, and Asia Rolls, who will join Dietmar and Kazuyo, who will still stay in the organizing committee because they join later. And we are absolutely delighted to have their wonderful ideas and effort to bring these global IMMUNOTALKs to all of you. So in the very last slide, I let my friend Elina Soniga bring to you what our organizers and all of us have prepared. So I will now move to the last slide.
Okay, and so I don't think that words are needed. This is a spectacular program, right, with amazing speakers. And, we cannot wait to keep learning about immunology and keep bringing immunology to every corner of the world. Whoever wants to learn can just connect live or via YouTube, right? So
Absolutely.
I think that with that, Carly, we will close and then we will distribute this via social media, but we definitely ask everyone to help us spreading the word, right? And so the more the merrier. And so if you could distribute the flyer in the that will be in social media, in your department, with your friends, amplify, that will be very much appreciated.
Thanks, everybody. Have a great end of the year, a wonderful start of 2024. Thank you, team, for such a wonderful talk to finish this series. Thank you, Sue. We are very in debt to all your wonderful, wonderful contributions and to all the organizers. Thanks.